Hello everyone, my name is Jeannette Bouchard and I am an infectious diseases pharmacist at WakeMed Health and Hospital System. In this presentation, I will be reviewing the pertinent drug information regarding the SARS-CoV-2 viral vector vaccines. In this video, we will be discussing the SARS-CoV-2 vaccines utilizing viral vector technology. Currently, there are 40 vaccines utilizing viral vector technology in preclinical and 17 in clinical development. The vaccines seen on the slide are the 4 that have current phase 3 clinical trials underway. These will be the focus of the current video and future updates. AZD1222 from AstraZeneca was originally studied as a 2-dose regimen 28 days apart. However, longer intervals in between prime and boost are approved for use in certain areas of the world. It should be stored at 2 to 8 degrees Celsius, and currently its projected costs are $3 to $4 per dose. AZD26AZD1222 from Johnson & Johnson is currently being studied as a 1 and 2 dose regimen 56 days apart. This vaccine is stable for 2 years at -20 degrees Celsius and 3 months at 2 to 8 degrees Celsius and set to cost about $10 per dose. Recombinant adenovirus 26S is also being studied in combination with recombinant adenovirus 5S in Russia. This prime boost combination is being studied with a 21-day interval. It is currently being manufactured as 2 different formulations, a liquid, which is stored at -18 degrees Celsius, and freeze-dried, which is stored at 2 to 8 degrees Celsius. The cost will likely be $10 per dose. Then as of December 29, 2020, a phase 2 open-label study in adults was initiated looking at AZD1222 in combination with recombinant adenovirus 26S. Viral vector vaccines rely on another virus that is engineered to express the desired antigen. In SARS-CoV-2's case, this would be the spike protein. Depending on the virus type, it is either designed to be non-replicating or replicating. Non-replicating vector vaccines are not able to make new viral particles and only produce antigen, whereas replicating vector vaccines produce new particles in the cell it infects, which will then go on to infect new cells and make antigen. The current SARS-CoV-2 vaccines use non-replicating vector vaccine technology. Research on these vectors has been conducted since the 1980s. Numerous viruses have been utilized as the basis for constructing these vaccines, including adenoviruses, parvoviruses, and togaviruses. Adenovirus vectors are one of the most utilized vectors and is the vector of choice in some SARS-CoV-2 vaccine candidates. Adenovirus vectors are ideal because they are able to stably support the expression of most target antigens as well as multi-pathogen vaccines. These vectors can also be produced in systems that support a high viral yield at low production costs, a trait that is important when creating a vaccine during a pandemic. Viral vector vaccines generally produce a strong immune response and do not need additional adjuvants like protein subunit vaccines would. When tested in combination with immune-stimulating components, no added immunity was seen. Adenovirus vectors specifically are able to produce a potent CD8 T cell and antibody response. One downside to adenovirus vectors is the pre-existing immunity to the virus in humans that may inactivate the viral vector. This is why we seek vaccine candidates with vectors, such as the chimpanzee adenovirus, to ensure that there is limited to no pre-existing immunity to these vectors. In relation to viral vector vaccines, especially those currently being created for SARS-CoV-2, the prime boost strategy of vaccination is common. There are currently 2 boosting mechanisms being utilized for SARS-CoV-2, homologous prime boost and heterogeneous prime boost. Viral vector vaccines are excellent at eliciting both humoral and cell-mediated immune responses, which can be optimized by boosting the initial vaccine. Traditional vaccines, such as inactivated and subunit vaccines, are not very effective in eliciting T-cell responses. When using a homologous boosting method, there can be formation of an immune response to the target antigen and the viral vector itself, thus blunting some of the boost vaccination potential and reducing the overall effectiveness of the vaccination series. In 1992, a landmark science report was among the first to utilize a heterologous prime boost immunization technique in a non-human primate model. The theory with a heterologous prime boost immunization technique is that by utilizing a different delivery system, a successful induction of both humoral and cell-mediated immunity can happen, such as utilizing a subunit DNA combination. With viral vector vaccines, because they induce both humoral and cell-mediated immunity better than other vaccine delivery systems, this is less of an issue. By utilizing a different vector with the boost, an avoidance of vector cross-reactivity can happen and lead to a more pronounced boosting effect. First, we will review the preclinical phase 1 and phase 2 data for AstraZeneca's AZD1222. The study breakdown of AZD1222 is represented here. Currently, there are 4 trials underway. COV-001, which is a single-blind phase 1-2 clinical trial out of the UK, will be discussed in more detail over the next few slides. But it was originally planned as a single-dose trial, however, only 88 patients still remain in the single-dose group of the study. COV-002, which is a single-blind phase 2-3 study out of the UK, is a study evaluating 2 separate dosing groups for AZD1222. A low dose, prime defined as 2.2 times 10 to the 10th viral particles, with a standard dose booster defined as 5 times 10 to the 10th viral particles, or 2 standard doses for the prime and booster doses. This low-dose regimen was not intentional at the start of the study and instead was a result of differences in quantification methods between manufacturing sites. It was decided that the qPCR reading of the dose was more accurate than the use of spectrophotometry, and all subsequent doses were adjusted based on qPCR for patients. Importantly, of the patients who received the low-dose standard dose regimen, none were greater than 56 years old. Enrollment for these participants began after the June 2020 amendment to use the qPCR-based doses. COV-003 was a single-blind phase 3 study out of Brazil that is evaluating a 2 standard dose regimen given up to 12 weeks apart with a target revaccination of 4 weeks. And COV-005 is a double-blind phase 1-2 study out of South Africa that will be evaluating a standard 2-dose regimen 4 weeks apart. This study will eventually have immunogenicity cohort for those living with HIV. These results are not available at the time of this video. A total of 1,077 participants were enrolled in COV-1, with 543 assigned to AZD1222 and 534 assigned to control. In the AZD1222 group, antibodies against spike protein peaked by day 28 and persisted until day 56 in the single-dose group. The booster dose group saw a similar antibody effect, but an increased median at day 56, 119 ELISA units in the single-dose group versus 639 ELISA units in the booster. Neutralizing antibodies were exhibited in all participants after a booster dose. At baseline, there were participants with high levels of neutralizing antibodies, and when vaccinated, these antibodies were boosted, showing a potential benefit in patients who may have been previously exposed to the virus. Next, we will discuss the pivotal efficacy trial for AZD1222. Boise and colleagues recently published an analysis from the 4 ongoing blinded randomized controlled trials described earlier in this video. Participants were ages 18 and older and were randomly assigned in a 1-to-1 ratio to receive either AZD1222 or control, which depending on the study site could have been saline or the meningococcal vaccine previously described in COV-1. There was a small subset in COV-2 who received a half dose as their first dose of the 2-dose regimen. However, the rest of the participants in the study received 2 standard doses of vaccine that were targeted towards a dose of 4 weeks apart. However, there were large intervals when the dose could not be given. For COV-2, for instance, 4 to 6 weeks was sufficient, and for COV-3, up to 12 weeks was allowed. 53.2% of participants in COV-2 in the low-dose standard dose group received a second dose at least 12 weeks after the first, and only 0.8% received a second dose within 8 weeks of the first. The primary objective was to evaluate the efficacy of AZD1222 against symptomatic COVID-19. This was defined as positive serology in at least one qualifying symptom. COV-5 was the only study that addressed events more than 14 days after booster as part of their primary outcome. However, the primary outcome in the pooled analyses specifically addressed infection more than 14 days after the second dose of vaccine. Secondary efficacy endpoints include severe COVID-19 disease, hospital admission associated with COVID-19, ICU admission associated with COVID-19, and deaths associated with COVID-19. Antibodies against SARS-CoV-2 non-spike proteins were also assessed in the secondary efficacy endpoints. There was a total of 23,848 participants recruited and vaccinated across all 4 studies. 1,077 in COV-001, 10,673 in COV-002, 10,002 in COV-003, and 2,096 in COV-005. A total of 11,363 participants in COV-002 and 003 met the inclusion criteria for the primary analysis. Of those included in the primary analysis, 2,741 participants from COV-002 received the low-dose standard dose, 1,367 of which actually received the treatment low-dose standard dose regimen. The other 1,374 received the meningococcal vaccine. Baseline demographics were broken down based on study and are seen here on the slide. A majority of participants were female and white. There was also a high percentage of health and social care workers. Regarding comorbidity breakdown, around 10% of participants in each study had cardiovascular disease and around 10% had respiratory disease, with very few having diabetes, between 1 and 3% depending on the study. Notably, COV-002, which is the only study to include low-dose standard dose regimen, only enrolled participants between the ages of 18 and 55 years old, and overall, across all studies, there was a limited number of participants enrolled above 56, and even less so above 70 years old. Of the approximate 11,000 participants included in the primary efficacy analyses, there were 30 cases in the vaccine arm and 101 cases in the control arm, resulting in a vaccine efficacy of 70.4%. For participants who had any serologic COVID-19 test result, there were 68 cases in the vaccine arm and 153 in the control arm, which equates to a vaccine efficacy of 55.7%. Additionally, certain studies were designed to assess asymptomatic disease in participants. Of those that were included in this analysis, there were 29 cases in the vaccine arm and 40 in the control arm, resulting in a vaccine efficacy of 27.3%. For the secondary efficacy endpoint of hospitalized patients, there were 10 participants hospitalized more than 21 days after their first dose of vaccine. 2 of these were considered severe COVID-19 cases, and there was 1 fatal case. All cases that resulted in hospitalization or severe COVID-19 and death were in the control group. Preprint data was released that reports a more detailed analysis of AZD1222's single-dose efficacy, as well as the impact of the timing of the second dose. Additional cases from an extra month of follow-up were included. A total of 17,177 participants were included in the efficacy analysis, 8,597 in the AZD1222 group and 8,580 in the control group, with a total of 332 cases of primary symptomatic COVID-19 occurring more than 14 days after a booster dose. A single standard dose of vaccine provided protection against primary symptomatic COVID-19 within the first 90 days, with a vaccine efficacy of 73% between 31 and 60 days, and 78% between 61 and 90 days. Overall, for any positive PCR from 22 to 90 days following a single standard dose vaccine, efficacy was 67%. Based on the current data, efficacy seems to have no significant impact within the first 90 days. However, there are currently no data for longer than this, and this is the reason for the continued recommendation to get a second dose of the vaccine, albeit a longer interval could be appropriate. When addressing the effect of redosing intervals on AZD1222, the preliminary analysis from December saw that vaccine efficacy was higher after the second dose with those who had a longer prime boost interval. It also saw better vaccine efficacy in those that had the low-dose standard dose regimen, which was addressed in previous slides. In the additional analysis, vaccine efficacy was still higher in those with longer intervals, with an efficacy of 82.4% in all doses and a dosing interval of 12 weeks or more. Point estimates of efficacy were lower with shorter intervals. However, these should be interpreted cautiously as some confidence intervals overlap. Higher binding and neutralizing antibody titers were also observed in sera at longer prime boost intervals, as shown by the graphs, indicating a potential relationship between humoral responses and vaccine efficacy. Although these data are based on a limited number of participants and may not represent the facts of a full data set. The data on the previous and current slides support the emergency use authorization for AZD1222 discussed on the next slide. A standard dose regimen is preferred operationally and likely had a lower efficacy than low dose regimens due to the dosing interval, and this had less to do with the dose itself. For key safety outcomes noted in the pooled analyses, local and systemic reactogenicity appeared to be lower and less intense in older adults, the low-dose regimen, and after the second dose. Serious adverse events and adverse events of special interest were balanced between vaccine and control arms in the analyses. Of the 175 events reported, 3 were considered possibly related to either the treatment or the control group. One of these events was a case of hemolytic anemia in the control group. The second was transverse myelitis in the vaccine group, and the third was a fever of greater than 40 degrees Celsius that was reported 2 days following vaccination. It is not known which group this participant fell into, but they received the second dose without a subsequent reaction. There were 2 additional cases of transverse myelitis noted during these trials that led to a temporary pause of the clinical trials for AZD1222. These 2 were later deemed to be unrelated to the vaccine by an independent committee of neurologic experts. Regarding deaths, so far in the pooled analyses, there were 4 non-COVID related deaths across all studies, 3 in the control group and 1 in the vaccine. All were considered unrelated to the study. In summary, based on current data from both preclinical and clinical trials, AZD1222 is safe and effective against COVID-19 with 70.4% vaccine efficacy. Serious adverse events were low and consistent between treatment and placebo arms. Contrary to the mRNA vaccine candidates described in a different video, local and systemic reactions following the second dose were less intense. Although they were less intense, it is still important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people, such as healthcare workers. Systemic symptoms can overlap with many of the currently utilized symptom screening tools for workplaces. Encouraging vaccination at the end of the work week may be appropriate to avoid this overlap. This analysis pooled studies from a variety of sites globally, including Brazil, South Africa, and the UK. Additionally, it included a large proportion of participants who were at a particularly high risk of contracting disease based on their occupations. Although limited, there was also data on AZD1222's effect on asymptomatic disease. As these trials continue, this will be an important factor to consider when evaluating differences between the available vaccines on the market. Of all these factors, perhaps one of the most important aspects of this vaccine is the cost per dose relative to the other vaccine candidates, $3 to $4 per dose versus $20 per dose, and the easy storage aspect as it only requires refrigeration. In terms of what is missing from the data, the biggest data point that is missing is the optimal dosing regimen and interval. Based on the data in the pooled analyses, the accidental lower dosing regimen of AZD1222 seems promising in comparison to the efficacy outcomes for the standard dosing regimen. Although this must be interpreted cautiously, as the lower dosing regimen was only given to a limited number of participants in one study, and it did not include older participants. The low-dose regimen also had more participants receive their second dose at a longer time interval in between, which may also contribute to the differences in efficacy. Currently, AZD1222 has a temporary approval by the UK Department of Health and Social Care. Under this approval, AZD1222 is dosed at the standard dose of 5 × 10 to the 10th viral particles, or 0.5 ml, and is approved to be redosed between weeks 4 and 12 after the first dose. Other major gaps for the general population are the long-term safety and efficacy data, as well as the vaccine's efficacy against severe disease. There were also too few cases of severe disease to be conclusive about any kind of overwhelming efficacy for prevention of severe disease or hospitalization. Further data will be needed to address these issues, and until then, continued public health reminders such as mask wearing and social distancing is strongly encouraged. Further data is also needed in specifically excluded populations such as pregnancy and breastfeeding and immunocompromised. There is currently no data to speak to safety and efficacy in these populations. As more data from clinical trials and patients receiving the vaccine via EUA access, we will hopefully have more information to speak to these populations. AZD1222 was given authorization for temporary supply by the UK Department of Health and Social Care and the Medicines and Healthcare Products Regulatory Agency. This information here is the guidance set out by the UK for use of this vaccine. The approved dose is the standard dosing regimen, and it should be given within 4 to 12 weeks of the first dose. It is approved for ages 18 and older with refrigeration storage requirements between 2 and 8 degrees Celsius for a 6-month expiration. Once the vial is punctured, it must be used within 6 hours. It will also come as a 4 and 5 milliliter vial with 8 and 10 doses available, respectively. The vaccine does not have any preservatives in it, and the guidance states that there are currently no data to this vaccine in persons who have previously received a full or partial vaccine series with another COVID-19 vaccine. Next, we will review the preclinical Phase 1 and Phase 2 data for Johnson & Johnson's AZD26AZD1222. Vaccines delivered by adenovirus type 26 vectors or AZD26 vectors have been shown to be safe and effective in humans with the ability to induce both humoral and cell-mediated immunity. The AZD26 vector delivery system has been used in the Ebola vaccine, currently approved in Europe, and in RSV. As of March 2020, AZD26-based vaccines were administered to approximately 67,000 study participants, most of these being from the Ebola vaccine study in the Democratic Republic of Congo. In a multi-center, placebo-controlled Phase 1/2a study in adults, AZD26AZD1222 was evaluated for safety, reactogenicity, and immunogenicity in 2 different dosing levels and in 2 different dosing strategies. The vaccine was studied at a lower dose of 5 × 10 to the 10th as a single or 2-dose regimen, and it was studied at a higher dose of 1 × 10 to the 11th viral particles as a single or 2-dose regimen. Cohorts 1 and 3 were randomly assigned to a 1:1:1:1:1 ratio as shown on the slide. Data in the study were collected after the administration of the second dose in Cohort 1A, with a median follow-up time of 94 days, and after the first dose in Cohort 3, with a median follow-up time of 30 days. Cohort 2 will collect data and be designed to assess the longer-term data of a single-dose regimen with a 2-dose regimen, as well as the safety and immunogenicity of a single booster vaccination 6, 12, or 24 months after completion of the primary regimen. This data is not presented here in the study overview. Due to initial immunogenicity and safety data suggesting a single dose of AZD26AZD1222 at the lower dose induces an immune response that meets pre-specified criteria, this is the dose selected for the Cohort 2 of the Phase 1/2 studies. A total of 405 patients were enrolled for Cohort 1 and 402 received the first dose. A total of 405 were enrolled in Cohort 3 and 403 received the first dose. At baseline, only 2% of patients were seropositive for SARS-CoV-2 in Cohort 1 and 1% in Cohort 3. Patients were excluded if they had a history of malignancy within 5 years, abnormal immune function, were pregnant, breastfeeding, or planning to become pregnant during the study period, and were not allowed to have comorbidities that increased risk of severe COVID-19 for cohort 3, with the exception of mild hypertension. Baseline characteristics were similar across groups in both cohorts. At baseline, the binding antibody geometric mean concentration, or GMC, values in all participants were lower than the lower limit of quantitation. In cohort 1A, by day 29, the values had increased to 478 in the low-dose placebo and 586 in the low-dose group, 625 in the high-dose placebo group, and 788 in the high-dose, with seroconversion of 99% or more in all groups. These all further increased by day 57. In cohort 3, the GMCs in all participants at baseline were again lower than the limit of quantitation. By day 29, GMCs were 312 in the low-dose group, 350 in the high-dose group, with 96% seroconversion. The neutralizing antibody titer, measured as the geometric mean titer, or GMT, is shown on the slide. In cohort 1A, at day 57, GMT were 310 for the low-dose placebo group, 288 for the low-dose, 370 for the high-dose placebo, and 488 for the high-dose group. Incidence for seroconversion was 96% in all groups, except the low-dose placebo, which had 100% conversion. GMTs for cohort 1A 14 days after the second dose were 827 in the low-dose and 1,266 in the high-dose groups. GMTs for cohort 3 were numerically similar at day 29 as cohort 1, with a GMT of 277 in the low-dose and 212 in the high-dose groups. Regarding S-specific T-cell responses, a low-dose recipient in cohort 1A and high-dose recipient in cohort 3 had measurable Th2 responses. However, the ratio of Th1 to Th2 was well above 1, suggesting a skewed Th1 response in the individuals with measurable Th2. Next, we will review the phase 3 data for AZD26CoV-2S. The phase 3 ensemble data was presented and represented a cutoff point of January 22, 2021 for primary efficacy and safety analysis. The phase 3 study is an ongoing, randomized, double-blind, placebo-controlled study to evaluate the safety, efficacy, and immunogenicity of AZD26CoV-2S administered as a single dose in adults greater than 18 years old. The target enrollment for this study is 40,000 participants with a 1-to-1 randomization to vaccine or placebo. The study goals were to have at least 30% of the enrollment be older than 60 years old, with only 20% of the enrollment aged 18 to 40 years. So an emphasis on participants who are 40 years or older. AZD26CoV-2S was given as a one-time dose of 5 times 10 to the 10th viral particles, or 0.5 milliliter injection. A staged enrollment strategy was completed for the protocol, starting with participants between the ages of 18 and 60 without comorbidities, and ending with participants greater than or equal to 60 years old with and without comorbidities. Participants who were excluded included those who were pregnant or planning to become pregnant within 3 months of randomization, and participants with abnormal immune function, such as those on chronic steroids or autoimmune disease. The co-primary endpoints were efficacy of a single dose of vaccine to prevent centrally confirmed moderate to severe critical COVID-19 occurring at least 14 days after vaccination and at least 28 days after vaccination in study participants without evidence of prior SARS-CoV-2 infection at baseline. Secondary endpoints included vaccine efficacy to prevent or vaccine impact on severe critical COVID-19, COVID-19 requiring medical intervention, COVID-19 related death, any symptomatic COVID-19, asymptomatic COVID-19 inferred via seroconversion, and COVID-19 per FDA case definition. Safety assessments included solicited local and systemic adverse reactions in the 7 days following vaccination and unsolicited, medically attended, and serious adverse events following vaccination. There was a total of 43,783 participants with a documented study vaccine administration. 21,895 received AZD26CoV-2S, and 21,888 received placebo. Full analysis set was defined as all randomized participants with a documented study vaccine administration. This was used for all safety analyses except solicited adverse events. The per-protocol set was defined as all participants in the full analysis set who had no immunologic or virologic evidence of prior COVID-19 at the time of vaccination and no major protocol deviations. In the full analysis set, 45% of participants were female and 19.6% were older than 65 years of age. Overall, 58.7% of participants were white, 9.4% were Black or African American, 9.5% American Indian or Alaskan Native, and 5.6% were mixed race. 45.3% of participants were Hispanic or Latino. At least one comorbidity was present for 40.8% of participants, with the most common comorbidity being obesity, with 28.5% of the population. Baseline demographics are broken down on the slide and were similar across both groups. There were 9.6% of participants in the full analysis set that were SARS-CoV-2 seropositive at baseline, and these participants were not included in the per-protocol set and primary endpoint analyses. Of note, well-controlled HIV was included in this trial, and there were 1,218 participants with HIV, or 2.8%. The primary efficacy analysis in the VRBPAC briefing document was based on the per-protocol set, which consists of vaccinated participants who were seronegative at baseline and whose cases were centrally confirmed. For the first primary endpoint, moderate to severe or critical COVID-19 at least 14 days after vaccination, there were 116 cases in the treatment arm and 348 in the placebo arm, with a vaccine efficacy of 66.9%. For the second primary endpoint, moderate to severe or critical COVID-19 at least 28 days after vaccination, there were 66 cases in the treatment arm and 193 in the placebo arm, with a vaccine efficacy of 66.1%. The slide shows the interim dataset cumulative incidence rate of time to moderate or severe or critical COVID-19 starting 1 day after vaccination with the full analysis set. The blue line represents AD26-CoV-2S and the red line represents placebo. For the secondary endpoint of severe or critical COVID-19 within 14 and 28 days, as of the cutoff date, there were 74 and 39 centrally confirmed severe or critical COVID-19 cases, respectively. Vaccine efficacy for the two time points were similar. Efficacy seems greater when cases before 28 days are excluded. However, confidence intervals for both are overlapping. When looking at COVID-19 requiring medical intervention with onset at least 28 days after vaccination, the vaccine efficacy was 100%. Small numbers make this difficult to interpret since the confidence interval is very wide, but overall, this is encouraging data for this COVID-19 vaccine. Interestingly, this study had endpoints to specifically address asymptomatic COVID-19 via SARS-CoV-2 RT-PCR, or molecular test from any available respiratory tract sample during preset times, day 1, day 29, and day 71. Asymptomatic testing after day 29 showed 10 cases in the treatment arm and 37 in the placebo arm, with a vaccine efficacy of 74.2%. There were 7 COVID-19 related deaths reported in the study as of February 5, 2021, and all 7 were in the placebo group in South Africa. Subgroup analyses of the primary endpoint were conducted to evaluate vaccine efficacy by risk factor for severe COVID-19. Vaccine efficacy against moderate to severe or critical COVID-19 with onset at least 28 days after vaccination was lower for participants with comorbid conditions and those who were greater than or equal to 60 years old. Confidence intervals for most subgroup analyses, including those with comorbidities and of older age, are wide, likely due to the short follow-up time for these particular subgroups. The majority of COVID-19 cases were among participants in the United States, South Africa, and Brazil. There was lower efficacy observed, specifically in South Africa, when compared to the United States cases, although these confidence intervals also overlap quite a bit and these numbers are difficult to interpret. In the United States, 73.5% of cases were sequenced and 96.4% were the Wuhan H1 variant D614G. In South Africa, 66.9% of cases were sequenced, of which 94.5% were identified as the B1351 variant. In Brazil, 69.3% of cases were sequenced, and 69.4% of those were identified as the P2 lineage variant. The rest were identified as the Wuhan H1 variant D614G. Vaccine efficacy against specific variants cannot be fully evaluated based on the presented data due to selection bias involved with prioritizing cases to be sequenced first, and only some of the cases fully sequenced as of the preliminary analysis. Overall, participants with medically attended adverse events, serious adverse events, and deaths were balanced between groups. Rates of unsolicited adverse events were also balanced across treatment groups, although there was a slightly larger percentage of unsolicited adverse events noted in the vaccine group that were related to the study product, specifically for events that are related to solicited adverse events such as chills, fatigue, and vaccine site pain. A greater percentage of solicited adverse reactions were reported in the vaccine group compared to placebo. Rates of adverse reactions were lower in participants greater than or equal to 60 years old compared to younger participants as seen on the slide. The most frequently reported local adverse reaction was injection site pain, followed by erythema and then swelling. Overall, the rate of local adverse reactions were similar between those who were seronegative versus seropositive for SARS-CoV-2 at baseline. The median time to onset of local adverse reactions was within 2 days of vaccination, and the median duration was 2 days for erythema and pain. The most frequently reported solicited systemic reactions were headache and fatigue. The most frequent grade 3 systemic reactions were fatigue and myalgia, although these grade 3 reactions were reported infrequently. The overall rate of systemic adverse reactions was similar in vaccine recipients who were seronegative for SARS-CoV-2 and those who were seropositive at baseline. The median time to onset for all solicited systemic adverse reactions was within 2 days of vaccination, with median durations being 2 days for fatigue, headache, and myalgia, and 1 day for nausea and fever. In summary, based on current data from both preclinical and clinical trial data, AZD1222 is safe and effective against COVID-19 with 66.9% vaccine efficacy at least 14 days after vaccination. Serious adverse events were low and consistent between treatment and placebo arms. Contrary to the mRNA vaccine candidates described in a different video, local and systemic reactions were less intense. Although they were less intense, it is still important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people such as healthcare workers. Systemic symptoms can overlap with many of the currently utilized symptom screening tools for workplaces. Encouraging vaccination at the end of the work may be appropriate to avoid overlap.Although limited, there are some data on AZD1222's effect on asymptomatic disease. As these trials continue, this will be an important factor to consider when evaluating differences between the available vaccines on the market. Of all these factors, perhaps one of the most important aspects of this vaccine is the cost per dose relative to the other vaccine candidates, $10 per dose versus $20. And the easy storage aspect, as this vaccine is stable for 2 years at -20 degrees Celsius and 3 months at 2 to 8 degrees Celsius. In terms of what is currently missing from the data, the long-term safety and efficacy data, as well as the vaccine's efficacy against severe disease and clinical efficacy against variants. There were too few cases of severe disease to be conclusive about any kind of overwhelming efficacy for prevention of severe disease or hospitalization. However, the current data presented here is very encouraging and is consistent with other vaccine platforms currently EUA approved in the United States. Further data will be needed to address these issues, and until then, continued public health reminders such as mask wearing, social distancing are strongly encouraged. This phase 3 data takes an important step when addressing clinical efficacy against the current variants that are predominant in Brazil and South Africa. Unfortunately, no conclusions can be made on the current data due to missing data points and biased approach to case sequencing. Further data is also needed in specifically excluded populations such as pregnancy, breastfeeding, and immunocompromised. There are currently no data available to speak to safety and efficacy in these populations. As more data becomes available, patients receiving the vaccine via EUA access, as well as clinical trial data, will hopefully drive more information to speak about these populations. In light of the newly classified variants of SARS-CoV-2, there are concerns that currently available vaccine therapies may be less effective. There are now numerous variants of concern and interest addressed by the CDC. Currently, there are data with viral vector vaccines and 2 of the variants of concern, SARS-CoV-2 B117, which was first noted in the UK, and SARS-CoV-2 B1351, which emerged in South Africa. These variants seem to spread more easily and quickly than other variants, which may lead to more cases of COVID-19. Recent data for AZD1222 against some of these variants has become available. Although data for the clinical efficacy of AZD1222 with these variants is limited and less robust than data for non-variant strains. Amari and colleagues conducted a study of AZD1222 efficacy for the B117 variant compared to non-B117 variants, shown on the slide as the Victoria strain. They found that viral neutralization activity was 9-fold lower against the B117 strain, but that vaccine efficacy against symptomatic COVID-19 positivity was similar between strains. Madhi and colleagues conducted a multi-center, double-blind, placebo-controlled trial in South Africa where participants were given AZD1222 in a 2-dose regimen 21 to 35 days apart. This is the COVE-005 study. They found that the vaccine efficacy was 10.4%, specifically with the B1351 strain, which was 92.9% of their primary endpoint cases. And when compared to cases earlier in the trial time period, prior to determination of the B1351 strain, the efficacy was higher at 75.4%, with a 95% confidence interval of 8.9 to 95.5. In addition to the data for AZD1222, AD26 CoV-2 had a study site in South Africa. Of the patients who were COVID positive and had their virus sequenced, 94.5% were the B1351 variant. The vaccine efficacy for South Africa participants were 64%, which was slightly lower than the point estimate when compared to the US participants, but confidence intervals overlapped, suggesting the true efficacy may be similar. Further clinical data are needed to elucidate the efficacy of these vaccines with current SARS-CoV-2 variants. On March 18, 2021, the European Medicines Agency announced that a rare thrombosis with thrombocytopenia syndrome was noted after receipt of AZD1222. Cerebral venous sinus thrombosis, or CVST, was the manifestation of these rare events. A mechanism similar to heparin-induced thrombocytopenia has been the current explanation for the occurrence. Following this announcement, 6 cases of CVST with thrombocytopenia following AD26 CoV-2S was reported through the VAERS system and subject to being reviewed. On April 13th, the EUA was paused for AD26 CoV-2S by the FDA and the CDC. After review of these cases, on April 23rd, the CDC and FDA recommendation was reinstituted. This rare event has occurred primarily in younger women, those less than 40 years old, and in patients without diagnosed thrombophobia. The thrombocytopenia in most cases was profound with platelet nadir count of 19. Most cases also had a positive heparin PF4 hit antibody ELISA test. For patients in the US presenting with CVST following AD26 CoV-2S, symptoms began at least 6 days after vaccination, and patients presented to the hospital around day 7 of symptoms. It is important to be aware of this exceedingly rare adverse event, but to also keep in mind the thrombosis risk in patients infected with COVID-19 and the benefits of vaccination with an extremely effective vaccine to prevent an infection that would put a patient at higher risk of thrombosis than the vaccine would. Next, we will discuss the phase 1 and 2 data of Russia's viral vector candidate Sputnik V, which is a combination series of an AD26 vectored vaccine and an AD5. 38 total participants were included in each study. 9 participants received either recombinant AD26 or recombinant AD5 in either the frozen or lyophilized vaccine formulation. The frozen formulation must be stored at -18 degrees Celsius and the other stored between 2 and 8 degrees Celsius. 20 participants received the prime and boost regimen of either formulation. In both studies, systemic reactions, local reactions, and changes in labs were the adverse events reported. The most common local reactions were pain at injection site, and the most common systemic reactions were hyperthermia, headache, and asthenia or fatigue. Most of these reactions were mild and transient in nature, including the changes in laboratory values. They were also more common after the second vaccination for participants receiving a 2-dose regimen. It is notable that in the lyophilized vaccine formulation, there was numerically lower reports for some local and systemic reactions compared to the frozen formulation, such as hyperthermia, headache, and asthenia or fatigue. None of the adverse events led to withdrawal from the study. Beginning at day 21, post either AD26 or AD5, receptor binding domain specific IgGs was detectable in 100% of participants. In phase 2, boosting with recombinant AD5 led to an increase in RBD specific IgG titers. An analysis of neutralizing antibodies to SARS-CoV-2 showed that only administration of both recombinant adenovirus 26 and 5 led to the production of 100% and neutralizing antibodies in participants, whereas administration of just AD26 led to seroconversion of only 61.1% of participants. Post-vaccination titers were significantly higher than convalescent plasma at days 28 and 42. In addition, cellular immune responses showed antigen-specific T helper and T killer cells with an increase in interferon gamma secretion. These responses are consistent with a Th1 biased cellular response towards vaccination. Additionally, this study addressed neutralizing antibodies to both viral vectors and found that recombinant AD26 did not neutralize recombinant AD5 when serum was analyzed, suggesting no cross-reactivity between vectors in a successful heterologous prime boost methodology. Next, we will discuss phase 3 data for Sputnik V. This was a phase 3 double-blind placebo-controlled trial at 25 hospitals in Russia. Participants were randomly assigned in a 3-to-1 ratio to receive vaccine or placebo. Healthy adults who were 18 years and older with no history of COVID-19 at randomization via PCR, IgM, or IgG were included. Key exclusion criteria were patients who were immunocompromised in the 3 months before enrollment, pregnant or breastfeeding, had acute coronary syndrome or stroke in the past year, neutropenia, or HIV. Participants were divided into 5 age strata: 19 to 30 years, 31 to 40 years, 41 to 50, and 51 to 60, and greater than 60 years. Participants were to receive either a prime boost regimen of 10 to the 11th viral particles of recombinant AD26, followed by 10 to the 11th particles of recombinant AD5 21 days later, or placebo. The primary outcome for the study were the proportion of participants with COVID-19 confirmed by PCR from day 21 after receiving the first dose. Secondary outcomes were the severity of COVID-19, changes in antibody levels against SARS-CoV-2 glycoprotein S, proportion of participants with antibodies against SARS-CoV-2 N protein, changes in neutralizing antibody titers, and changes in cellular immunity levels. Safety was assessed via local and systemic reactogenicity. Serious adverse events were quantified on the basis of requiring hospitalization. Of note, only the frozen formulation of the vaccine was utilized in this study. The preliminary analysis published included 14,964 participants in the vaccine group and 4,902 in the control group who received 2 doses at the time of database lock on November 24, 2020, and were included in the primary outcome analysis. 9,258 in the vaccine group and 3,038 in the placebo group were included in the general safety analysis. The immunogenicity analysis included a total of 456 participants for RBD-specific IgG analysis, 100 in neutralizing antibody analysis, and 58 in interferon analysis. Baseline characteristics for participants who received 2 doses were a mean age of 45.3 years in both vaccine and placebo groups, and the gender, incidence of concomitant diseases, and infection risks were similar between groups. Highlighted on the slide are the risk of infection categories utilized in the study. High risk denotes those whose work involves interaction with patients with a confirmed diagnosis of COVID-19. Medium risk were those who have a professional contact with a large number of people, and general or low risk were those with no additional risks associated with their professional activities. There was a fairly large number of low-risk participants in this study, which is different from other vaccine trials that were specifically enrolling patients who were higher risk, such as healthcare workers. The result of the primary outcome of the study saw 16 cases of COVID-19, 21 days after dose 1 of vaccine, and 62 cases following dose 1 of placebo, with a vaccine efficacy of 91.6%. This efficacy was consistent through most subgroups in all ages and genders. Notably, the efficacy was slightly higher for participants older than 60 years old, point estimate of 91.8%, although confidence intervals do overlap. When addressing the outcome most consistent with other vaccine trials, which is COVID-19 disease following dose 2, there were 13 cases in the vaccine group and 47 in the placebo group, with a vaccine efficacy of 91.1%. For secondary outcomes, the number of cases of moderate or severe COVID-19 were 0 in the vaccine group and 20 in the placebo group, with a vaccine efficacy of 100%. Moderate COVID-19 was defined as fever, respiratory rate greater than 22, shortness of breath during exertion, pneumonia on CT, oxygen saturation less than 95%, and CRP greater than 10. Of note, COVID-19 cases were detected through report of symptoms and follow-up PCR tests. Therefore, only symptomatic cases were included, and there are currently no data on asymptomatic cases. Other secondary outcomes regarding immunogenicity for the vaccine regimen were encouraging. There was a robust humoral response seen in all ages. Notably, there were 6 non-responders without any defined reason for non-response. Speculated reasoning was age, concomitant immunological disorders, or individual characteristics. The 2-dose regimen was able to induce a virus-neutralizing humoral response in the older patient population. The most common adverse events were flu-like illness, injection site reactions, headache, and asthenia or fatigue. Most of these adverse events were grade 1 or 2 in severity, with 0.38% being grade 3. Serious adverse events occurred in 68 patients with 70 episodes reported. 45 of these were in Sputnik 5 and 23 in placebo, making up about 0.3 or 0.4%. None of these serious adverse events were considered to be associated with the vaccine. 4 deaths did occur during the study at the time of analysis. 3 were with Sputnik 5 and 1 with placebo. 2 Sputnik 5 deaths were associated with COVID-19 and considered to be due to pre-existing cardiovascular and endocrinological conditions that were exacerbated by COVID-19, and the other was unrelated. In summary, based on current data from both preclinical and clinical trial data, Sputnik 5 is safe and effective against COVID-19 with 91.1% vaccine efficacy. Serious adverse events were low and consistent between treatment and placebo arms. In the phase 1-2 data, there seemed to be more local and systemic reactogenicity following the second dose of the regimen, which is fairly consistent with other frontrunner vaccine candidates. It is important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people such as healthcare workers. Systemic symptoms can overlap with many of the currently utilized symptom screening tools for workplaces. Encouraging vaccination at the end of the work week may be appropriate to avoid overlap. This study did stratify outcomes based on different age groups, and when the primary outcome was broken down between these age groups, there were similar outcomes, including for those who are older than 60 years old. These clinical outcomes were also consistent with the humoral immune responses measured. Although there are very small numbers of participants with immunogenicity data, and this should be interpreted cautiously. The primary outcome of this study was COVID-19 disease 21 days following dose 1 of the vaccine. This is different from the other vaccine trials that assessed COVID-19 following dose 2 of the series. Although efficacy was similar prior to dose 2 as it was after dose 2, suggesting early immunity is potentially achieved following the first dose, there is no explanation in the study for why this primary endpoint was chosen or if it was a post hoc decision which could potentially lower the reliability of the study. Other major gaps for the general population are the long-term safety and efficacy data, as well as the vaccine's efficacy against severe disease. There were too few cases of severe disease to be conclusive about any kind of overwhelming efficacy for prevention of severe disease or hospitalization. Further data will be needed to address these issues, and until then, continued public health reminders such as mask wearing and social distancing is strongly encouraged. Further data is also needed in specifically excluded populations such as pregnancy and breastfeeding and the immunocompromised. There are currently no data available to speak to safety and efficacy in these populations. As more data from clinical trials becomes available, we will hopefully have more information to speak to these populations. Specific resources to learn more about vaccine recommendations and frequently asked questions are listed here on this slide. The CDC provides excellent guidance for both healthcare providers and patients receiving vaccine, as well as separate information for the EUA-approved vaccines available. Thank you very much for watching. I hope this was a helpful summary of the SARS-CoV-2 viral vector vaccines. Again, this video will continue to be updated as information becomes available. If you have any questions regarding the material presented in this video, please reach out.